Hello everyone, my name is Prajakta Dhage and I am from Department of Zoology KTHM College Nashik. Today I'll be taking a lecture for MSc Part 2 Semester 3. The subject is soft skills and we are dealing with the second chapter, which is named as Unit 2. The topic is preparation of solution and this is the second lecture of the series. Preparation of solution part 2. We have already seen what is molarity, what is molality in the previous lecture, what is the normality. Okay, so today we will be dealing with some other concepts which are as follows. The first concept that we are going to see today is osmolarity. Now, in simple terms, osmolarity is the process of measurement of solute concentration. It is a number of osmoles of solute per liter of solution. Okay, so basically osmolarity is a number of solute particles per liter of the solvent. Usually it is written as osmoles per liter. This resultant number is of high importance as it allows the measurement of the osmotic pressure of a solution, and also it allows to determine how the solvent will diffuse across a semi-permeable membrane that is separating two solutions of different osmotic concentration. Okay, now the unit of measuring it is osmole. Okay, osmole is a non-SI unit of measurement that defines the number of moles of solute that contribute to the osmotic pressure of the solution. Okay, so osmole is non-SI unit and it is going to give you the number of moles of solute that are going to contribute to the osmotic pressure. It is measured using an osmometer. Now, what is the importance of this osmolarity? It is very important because cells cannot survive if the osmolarity of their surroundings are much different from their own. Okay, water moves from the side of the membrane to another side, that is from a lower to higher. In other words, it changes from the dilute side to the concentrated side. It is a continuous process in which dilute side loses water and becomes more concentrated, and the concentrated side gains water to become more dilute. Okay, so the cell survives by balancing water uptake and to become more dilute. Okay, so the cell survives by balancing water uptake and loss. Now we'll move on to what the difference that is there between osmolarity and molarity and osmolarity. Okay, now we have already seen in the previous video that molarity of a solution is the concentration of solute in that particular solution. Okay, it tells you how much of the solute is present. The unit of molarity is moles, whereas this osmolarity helps us to determine if water will move from one side of the membrane to the other side. Okay, that is, it means that whether the osmosis is going to occur or not will be described by the osmolarity. Okay, and we must note down the fact that water always moves across the membrane into the solution with higher osmolarity. Okay, so osmolarity can also be defined as the number of ionic species in molarity that has a characteristic range depending on the species examined. Okay, the maintenance of the cells, membrane vesicles or intercellular organelles like mitochondria, nuclei is dependent upon the transmembrane equilibrium of osmotic pressure. Okay, why is it so? In order to prevent the hypoosmotic or hyperosmotic stress. So, for example, cell culture media are usually adjusted to 290 to 320 milliosmoles to prevent any osmotic stress to the cell. If the osmolarity is not known, then a value could be determined by measuring the conductivity of the cells or the tissues homogenized in water if the plasma is not available. Clear? Now let's move on to another concept, which is osmolality. Okay. Now, osmolality of a solution refers to the concentration of osmotically active particles in that solution. Okay. So, it is number of solute particles in 1 kg of the solvent. Osmolality is a function only of the number of particles, right, and is not related to their molecular weight, size, shape or charge. So, the term osmolality refers to the number of particles of a solute per kilogram of solvent, as I have already mentioned. So, when considering the physiology of body fluids, the difference between the osmolality and osmolarity is negligible, because body fluids typically are dilute aqueous solution. Clear? So, a solution is said to be hyperosmotic, okay, if its osmolality is greater than that of the reference solution, okay, and hypoosmotic if its osmolality is less than that of the reference solution. So then what is osmotic solution? It is nothing but the solution in which osmolality is identical to that of the reference solution. Clear? Now, osmoles that do not cross the cell membranes freely, okay, are considered effective osmoles, okay, whereas those that do cross freely are termed as ineffective osmoles. So, you have effective osmoles and you have ineffective osmoles. Clear? The water permeable cell membrane is functionally impermeable to sodium and potassium. As a result, sodium and potassium molecules are effective osmoles and they exert osmotic pressure across the cell membrane. Okay? The net movement of water into or out of the cell is dictated by the osmotic pressure gradient. Osmotic pressure causes water molecules from an area of lower osmolality, that is higher water concentration, to move to an area of higher osmolality, which means lower water concentration, until the osmolalities of the compartments are equal. Clear? Now, we will move on to another concept, that is parts per million, that is ppm. Okay. Now, what is parts per million? Let's see. This is a way of expressing very dilute concentrations of the substances. Just as percent means out of 100, so parts per million or ppm means out of million. Usually, this describes the concentration of something in water or soil. 1 ppm is equivalent to 1 milligram of something per liter of water, or 1 milligram of something per kilogram of soil. Clear? Now any natural water contains dissolved chemicals, some of which are important human nutrients, while others can be harmful to human health. The concentration of water pollutant is commonly given in very small units such as parts per million or even parts per billion. For example, an arsenic concentration of 1 ppm means 1 part of arsenic per million parts of water. This is equivalent to 1 drop of arsenic in nearly 50 liters of water. Clear? Now let's move on to another concept, which is parts per billion. Now parts per billion is the number of unit of mass of any compound per billion units of the total mass. Okay, it is also given in terms of micrograms per liter. So ppb is used to measure the concentration of a say contaminant in soil or its sediments, and in that case 1 ppb equals to 1 microgram of substance per kg of solid. It's μg or micrograms per kg. Okay, now sometimes we use ppb to describe small concentrations in water. In this case 1 ppb is equivalent to 1 microgram per liter. Okay, now ppb is often used to describe the concentration of contaminants in air or say contaminants in water. Okay, so for example, let's say 1 ppb chlorine represents 1 part of chlorine in 1000 million parts or 1 billion parts of air by weight. Clear? Okay, so this is all from my side about the preparation of solution, and thank you so much.